Okay, well, thanks for this very nice introduction. First of all, I should have known better than to agree to speak after Sid Mukherjee. And then I'd like to issue two warnings. One to you. I'd like to dedicate this talk to the memory of a friend, Pierre Schönhammer. Pierre was designated about 7 or 8 years ago to be my official introducer in all my lectures. And he had, in 1 year, introduced me 3 or 4 times, which means he listened to me 3 or 4 times in 1 year, which is rather remarkable. And I was very pleased to see ID, his wife, here with us. Pierre passed away about a year ago. The second warning, I tend to walk around when I give lectures. And in Bangalore, I was giving a series of lectures, and I fall off the stage like every 5 minutes. And then this was on video, and it's on the net, and it's not my... So I would warn the people in the first row to beware. But the one good thing about it is that it keeps the audience really alert. They are there. No one fell asleep in Bangalore. Okay, so my talk has 4 parts, and the 4 parts reflect the title of the talk. And the 4th part, Jay, is my work, the hope. Because last time you heard me talk, asked, what have you done? Okay, so let me start with the introduction, and I'll tell you about the dawn of modern genomic research. It's very easy to pinpoint. On 2 consecutive days in February 2001, 2 papers were published, and they each contained a draft of the human genome. Our genome is a text. It's a text which is written on the DNA of every one of your cells. And this text is rather long. It has 3 billion letters. For comparison, note that the King James Authorized Version of the Bible, New and Old Testament combined, contains 1,000 of this number of letters. So it's an enormous text which is sitting on your DNA. And once this text has become known, this generated immediately 3 big things: big excitement, big data, and big hope for medicine. And these are the reasons, the 3 big reasons, that induced me to leave my comfortable existence as a theoretical physicist and plunge into the turbulent waters of computational biology. And there has been enormous progress since 2001, technological, computational, and scientific. To demonstrate the technological advance, note that it took 10 years for hundreds of dedicated researchers and $10 billion to come up with this first draft of the human genome in 2001. If any one of you is interested in getting the text, the 3 billion letters of your genome, if you want to do it today, this will cost you a few $1,000s, and it will be done about a day. So this is a tremendous advance. And we have at Weizmann a world-class support facility. It's called the Nancy and Stephen Grand Israel National Center for Personalized Medicine. I got it right. And this center builds exactly on these advances in the technology and on our computational skills in order to help us to generate advances in science. With all these great things and advances stated, we are hit every day by a question, a very nagging question. You have 14 years passed since the publication of this human genome. There have been 100,000 manuscripts published using genomic research, and 10 million man-years and countless of dollars invested. Where is the cure for cancer? So what we know is that there is no magic bullet. There is no antibiotics for cancer, and it is a poor man's version, what I'm going to tell you after you heard Sid Mukherjee, that cancer is not a single disease. There are the main reasons for the fact that there is no magic bullet are 3. Diversity. Any 2 persons who have breast cancer will have a slightly or substantially different disease. Heterogeneity, which is even more frustrating. If you cut out a tumor from a patient and you look at one piece of the tumor and then the opposite side of the tumor, you discover that you have 2 types of different malignant cells in the same tumor. So it's not just diverse, it's also very heterogeneous. And finally, it's a moving target. The cancer cells change. And if you find some therapy that seems to work and some cells survive this therapy, the cancer cells learn how to overcome the therapy that you hit them with, and therefore they are a moving target, which makes it extremely complicated to deal with it. And it's exactly this is the mission of the proposed Weizmann Integrated Cancer Center, to try to attack somehow this very complex and really apparently hopeless, but I hope not hopeless, problem. So having said all this, let me go to the next part of my talk. By the way, remember my talk has 4 parts, and as I go along, I'll try to tell you we are switching to the next part, so you know how long you still have to suffer. So here is now the next part of my talk, which talks about success. So there was enormous success in clinical applications of genomic research, and I ran a brief survey, a mini survey, asking some 4 or 5 oncologist friends from all over, from US, Europe, and Israel, and there are about 4 or 5 cancers for which there are standard genomic tests which are routinely performed in the clinic. And I list these here. The second is skin cancer, melanoma, and you will hear a lot about this from Ravid Straussman. And what I'll do, rather than going into detail of all these diseases, I'll look at the commonalities of these 4 success stories. And the first one is that all these treatments attack a known molecular target. They are targeted therapies. The second one is that the treatments target a subset of the patient. This is something you have to remember. This is a hard fact to digest, and we have to live with it. The treatments target only a subset of the tumors, and what's even worse, among those that are treated, only a subset actually respond to treatment. And these treatments mostly prolong life. They don't cure because the ability of cancer cells to develop resistance. And finally, surprisingly, all these 4 treatments address the first hallmark of the ones that were shown by Dr. Mukherjee. And I will use breast cancer as an example, and I will start going now one by one these commonalities and start with the first hallmark. This is uncontrolled growth. And to understand that, I'll just repeat what Sid already said, less eloquently than he did. Normal cells do not get up in the morning and say, let's divide. It's not a random decision. The decision to start dividing comes in response to signals that the cells receive. Now, you have to understand that you are a multicellular organism, and our cells talk to each other all the time and send signals. Just like I am talking now, I generate sound waves, which are density vibrations in the air, which reach your ears, which are detectors to detect this signal. And then you hear me, and the signal is processed in your brain, and you respond to the signal I sent you. In the same way, cells talk to each other. They do not use sound. They do not use radio waves. What they use is chemical signaling. And the same way as you have ears to detect sound, cells have receptors on their surface which stick out and wait for these chemical signaling molecules. So that's the language that cells use to talk to each other. Normal cells wait for growth signals in order to start dividing. And what cancer cells do, they are able to divide without any growth signal. It's like an army going ahead without its marching orders. So the question is, how do cells acquire this ability of growth without the marching orders? And this is the example I'm going to use. It's the particular kind of breast cancer. And the way they accomplish this is a very simple trick. A normal cell has 20,000 of this particular kind of receptor on its surface. A cancer cell will have 2 million of these receptors, 100-fold more. Imagine that you were sitting here, each one of you, not with 2 ears, but with 200 ears. And all 200 ears are tuned to identify the voice of Eytan Domany. And since you are now 100 times too sensitive, even the slightest noise of the microphone will be picked up and interpreted as, here I am harassing you again and again and again. So this is the trick that these HER2 positive cancer cells use to generate the ability of growth without growth signals. And I have a caricature here. This is the part of the receptor which is sticking out into out of the cell. And don't worry about this being too simplistic. Michal Neyman will show you a full movie of the process and go into a lot of detail about these receptors and how they activate the cell. So here is a mechanical analog. See, I'm a physicist. So this part which sticks out of the cell, its analog is the pedal of your accelerator in your car. This is the part of the engine system which is sticking into the driver's compartment. And normal cars accelerate in response to a stimulus. What's the stimulus? The pressure that your foot exerts on this pedal. Now imagine that the pedal is stuck. So stuck means that it's pushed down and remains pushed down. So now the car will accelerate irrespective of what you do with your foot. And this is exactly what happens with these cells. Now, how do we fight this? The remedy is a drug called Herceptin, which is a small molecule which binds to this receptor and inactivates it. This is the analog of the mechanic crawling in there and getting the pedal unstuck. So it's a simple remedy, and it works. And here I come to the next and what, in my mind, the most important aspect of this genomic treatment that you have to remember. I have drawn here 100 women, 100 breast cancer patients. And it turns out that only 20 of them have this form of cancer that are HER2 positive. The other 80, if you give them Herceptin, it doesn't matter. It's like giving them water, because they do not have this particular overexpressed molecule on their surface. So only 20 women will get Herceptin, a subset. Now here is another bad news story. It turns out that if you don't treat at all these 20 women, and let's say we set out our aims low, we want to keep these women disease-free for 4 years. So if you don't treat these women, 14 of them will not have recurrence of breast cancer, even if you don't treat them. So only 6 out of the 20 really need the treatment. And now here is the last part of the news. If you do treat them, only 3 out of these 6 will respond to therapy and go disease-free for 4 years. So now what do you say about this? So if you're a pessimist, you say, this is not a success story, because only 3 women benefited from this drug. Now, I'm an optimist, and I say Herceptin gave 3 women 4 disease-free years of good, high-quality life. And that's a major achievement. So just disappointment is a function of your expectations. So you have to set your aims at this level. You have to remember, because of the things I said, of the diversity of the disease, every drug that someone will come up with will be really effective only for a subset of the population of sick people. This is the rule rather than the exception. Okay, so I come now to the third part of my talk, which is frustration. And here I'll talk about breast cancer again. And when I first started to study and read about breast cancer, the aspect that struck me most was its incidence. It's amazing. It's 1 out of 8 women in the Western world has a bout with breast cancer during her lifetime. They don't die of it, but they encounter the disease. It's a major epidemic. And the treatment is fairly standard. Surgery, followed by radiotherapy, and then if there is some known target, then there is targeted therapy, like Herceptin. And then comes the major question, what to do next? Do we or don't we give this person systemic chemotherapy? And this chemotherapy is general. It is not targeted. It's not specific to the cancer cells. What it does, it's like carpet bombing. It destroys cells that divide. It's a poison. And the main problem with that is that normal cells also divide. Our hair grows. Our intestines, the internal lining of our intestines are replaced every 2 weeks. And all those cells, which have nothing to do with cancer, their division is part of our normal function in life, all those cells are also affected. And therefore, this type of treatment, its strength, that it's general, is also its weakness. That is, having this poison is detrimental to the patient's health. It affects her well-being, and it's also very expensive. And therefore, there is an acute need for prognostic and predictive indicators. We want to give systemic chemotherapy only to those women who really need it and really benefit from it. And we are looking for some way of taking material from the tumor at the time of resection, looking at it, and coming back with a suggestion to the clinician, should you or shouldn't you give chemotherapy? So in order to do that, this problem was really acute, and a whole large number of clinicians, biologists, and computational scientists attacked this problem. And the idea was very simple. Collect tumor samples from a large number of patients at surgery, freeze them, follow these women for years, and try to record for each woman whether her cancer was aggressive and how did she respond to therapy. And then measure on each of these tumor samples a very large number of numbers, 20,000, 30,000, 50,000 numbers for each tumor. So now, do it for a few hundred tumors and for a few hundred, well, let's stay with tumors, for a large number, few hundred tumors, and look for patterns. Try to identify in this zoo of tens of thousands of numbers some pattern which will separate aggressive tumors from less aggressive tumors, tumors that respond to chemotherapy from tumors that don't respond to chemotherapy. And try to do this and create a prognostic and predictive classifier. So this was the challenge, and I must say that so far this line of attack on the problem has failed. And this is the cause of the frustration, which is this part of my talk. So let me try to explain why. So this is a car. A car is made of 10,000 components, up to 12,000. I'm so sure that Maserati has more components than a Fiat. So, but this is what you see here is a car taken apart, and all its 10,000 nuts and bolts and belts are spread out. So the approach that we were taking to this problem is, I call it atomistic. Imagine that your car, something may be wrong with it, and you bring it to a garage, and what the mechanic does, it says, no problem. The car has 10,000 components, and I'll stick a thermometer to each of these components and end up with 10,000 temperatures, so each car will be characterized by 10,000 numbers. And I'll take a few hundred defective cars and a few hundred good cars, and I'll look for patterns that identify defective cars. Now, no mechanic works this way, as far as I know. And this is what we tried to do with breast cancer. And the most bothersome aspect of this attitude to the problem is that there was no knowledge used about cars. You look at these patterns, and it doesn't matter whether these are cars or space shuttles, airplanes, or what. And I advocate a different approach, and what I'll call a systems approach, and I was very pleased to see the last part of Sid Mukherjee's talk, which advocates, I think, the same view. A car is not just a collection of 10,000 components. A car has different systems, and here are a few. There are brakes, engine, transmission, cooling. Those of you who understand cars, look at this and immediately realize that the parts that I indicated as belong to the brakes don't belong to the brakes, and the cooling not to the cooling. So anyway, it's for demonstration purposes. Cars are made of systems, and what a good mechanic does, it looks at the car at a systems level. He looks and does some simple system-level test to test the brakes. And he ends up with a score, which says the dysfunctionality of the brakes is on a scale of 1 to 10, it's 6. So this way, what he does, he characterizes the car by a set of such system-level indicators. And if one wants to run a thorough test of the car, he will end up with on the order of 100 numbers, not 10,000, not 20,000, 100. But each number will have a meaning. It will not tell the mechanic the level of deregulation, of dysfunction, or defectiveness of that particular system. So what I advocate is to do the same with cells. Cells also have systems and biological processes which govern them and which they use. So rather than looking at all 20,000 molecules or a million molecules that constitute a cell, which is what we did before with the car, what I advocate is to do tests on the system level. And the only problem with this program is that cells are much, much, much more complicated than cars. And therefore, what works easily for a car is not so trivial to do for a cell. So let me show you the result of this. Okay, this is the outline of how I do the analysis. And the bottom line of it is, when I'm done, a cell has, say, a few hundred systems, which I know and I can recognize. And I end up with, for each tumor, with a set of few hundred numbers which score every system, just like brakes and so on. So the level at which I will do my analysis is system level, and I will end up and score every tumor over 100, 200, 300 systems. That's the program, and this is what comes out of it. So you look at this and say, what the hell? Now, so what do you see here? What you see is a collection of very large number of numbers. This is like an Excel file, and you may have seen those. It has 500 rows. Each row stands for a pathway, a biological process. And it has about 1,000 columns, and each column represents a particular sample, tumor. And now, if you look closely, you see that there is a patch here which is yellowish, and that patch sits on row 143 and column 789, and the color indicates the level at which pathway number 143 is defective in patient number 789. So you understand what this collection of numbers is. I cannot show you there are 500,000 numbers here. That's why I use this color coding. So you don't see much. You can stare at this for a week, for a month, and you won't make any sense of it. But then you can do a trick. You can rearrange the rows and the columns of this huge matrix. And that's what we have done. In this one thing of rearranging rows and columns to bring meaning out, we are the world champions. We is my group. And what you see here, it's hard to believe, but this is the same array of numbers. Sorry. This is the same array of numbers as that. All we did was reshuffling of the columns and the rows. And you see what beautiful structure comes out. And when we do that, we discover two new things that were not known. The first one is that there is a set of pathways which have to do with a particular kinase, one kind of molecule. And these pathways are very wildly deregulated in a subset of the breast cancer patients. About 70 out of 1,000 patients have a very red color here. And this very red color means that these pathways are very highly deregulated in these patients. It's a new class of breast cancer, and we are in the process of trying to figure out the clinical implications of this finding. And here is another one. There is one particular kind of breast cancer, which is called basal. It's about 150 of the 1,000 patients that have this type of cancer. It's an aggressive form of cancer, and it's one about which we know the least. Now, what we discovered, and here are the patients, between these black lines are the patients who have this kind of basal subtype. And it turns out that there is one type of pathways. All of them have to do with the immune system. And these pathways divide the basal tumors into two subclasses, one with a high level of deregulation of the immune system and one with a low level of deregulation. And to our surprise, we discovered that those that have a high level of deregulation of the immune system, which indicates high immune activity, those tumors have better outcome than the other half. And we can show this. What I show here is a curve which describes the probability of survival as a function of time. And if you have, not you, whoever, has this type of cancer, the one that has a high level of immune involvement, the outcome is better than if the patient has the low level of immune involvement. And this may have some direct clinical bearing for the basal patients, providing a simple biomarker to predict outcome. So this is more or less the end of my story. What have I done? The strategy I followed is a phenomenological or systems-based one. The rules are read the book, use existing knowledge, retain the essential aspects of the problem, and build on measurable quantities of features in your analysis. And there is a fourth one, which is use minimal models, which is very standard in physics. Always look for the simplest possible explanation for a complex phenomenon. And I found pretty problematic to get this fourth item across to my biologist friends. This is the one that they had difficulty with. So I found a movie to explain what physicists mean by a minimal model. Allow me to show you that movie. So I'll speak out if you cannot read the subtitles. Excuse me. Do you know what time it is? 10 past 10. Are you sure? Just as I said. Uh-huh. Thank you. Not convinced. Now he's coming back. Nice day. Yeah. Say, what time is it now? Again. 7:15. Damn, it's exactly 7:15. Unbelievable. How do you know what time it is by touching the donkey's balls? So I guarantee that you will all remember me as the guy who showed you the movie and I succeeded not to crash. Now, this is my support, and I would like to emphasize the first one, the Laird Charitable Foundation, which are donors, and they have been instrumental, and their support of my research has really changed my scientific life. And this is the last part, which shows you how special the Weizmann Institute is. I moved into biology about 14 years ago, and here you see members of the Weizmann faculty on the left side, biologists that worked with me. Among those that, Zeli Geshar is one of them, worked with me. I don't mean that we had coffee together. We wrote papers. We published papers together. So there are like more than 20 people here in 14 years. Amazing. We have one of the most interactive atmospheres that I have seen ever. And on the right side, in blue, you see the names of students and postdocs that worked with me during these years. We have the best people that I have seen ever, students, and I've been around. So I'm not saying that all our students are better than the students at Cornell and Harvard, but they are as good, and we are really fortunate to have this high quality of graduate students at Weizmann. We have a very, very thriving interdisciplinary atmosphere, which is really perfect for this kind of research that I've been talking to you about. Thank you very much.
So what a wonderful introduction as well to the Grand Center for personalized medicine. It really plays a role. The other point, the multidisciplinary approach that Weizmann is so proud of. We have a time for a couple of questions. Yes.
What was the person in the film able to tell you the date also, or was it just the?
I'm not sure I understand. What was?
Was the person in the video able to tell the date as well as the time, or just the date?
Could they tell the date as well as the time?
Oh,
Yes,
I'm
of
sorry.
course, you had
Yes,
to look,
yes.
had to look at the other direction.
Yeah.
No, in all seriousness, your diagram indicated one type of cancer. Are you looking across multiple cancers to get certain indications of
It's an excellent question. In what we are trying to do, I mean, it's an excellent question, and I'll try to answer it briefly, even though it deserves a long answer. I was telling you that even breast cancer is not a single disease, but 100 different diseases. And therefore, in order to come up with a predictor, I think one should really go into each subtype separately and analyze it separately, because the answer will be different.
Okay.
The method of prediction will be different for each subtype. What we have here is something that will work for basal tumors, not necessarily for the others. But right now I am sort of planning to go back to, I mean, this is the rule in biology. You solve one problem, and what I'm trying to go back to my physics training and to look for something universal. So I really plan to go through 20 different cancers and try to identify commonalities on this system-level treatment. So I don't know if I answered your question, but this is sort of the closest I can come to answering that. It was a very good question.
One more question, yes.
I'm sorry, please take the microphone so I can hear you.
You mentioned regulated and deregulated immune system. Deregulated, I understand, but how do you regulate?
Well, what I mentioned in this last example was the following. We take a pathway that belongs to the immune system, and we check to what extent the behavior of this pathway in the tumor is different from its behavior in normal breast. We call normal breast normal behavior. And if the behavior in the tumor is very different than in the normal tissue of the same kind, then we say this is deregulated. So it's not regulated and deregulated, but it's correctly regulated or normally regulated versus malignantly regulated. It was my bad terminology. I'm sorry about that.
Okay, thank you very much.